Good afternoon, students. Good afternoon, students. Today we'll be dealing with physiology of digestion. Now, today our speaker is Yatish Kumar sir. He has his kitty master's degree in zoology, microbiology, and also in education. Let us start with the physiology of digestion. See, in the previous class, we have dealt with about the structure of the digestion and also about the glands, different glands associated with it. Now, let us find out how this really works. That is, how the digestion takes place in all these chemical and mechanical processes. Yes, now I welcome Yatish Kumar sir. Please take over the class, sir.
Thank you, madam. Students, very good afternoon, all of you. So far, you have learnt in the zoology session about the alimentary canal and the digestive glands. Yes, in this session, we are going to discuss about the physiology of digestion. This topic is most important in the IBE examinations for 4 marks, as a long question for 8 marks also. So listen carefully and concentrate about the physiology of digestion. Now, students, look at the slide here. You are seeing different types of the food materials. Yes. So we are taking different types of the food materials every day. Now tell me why we are taking these food materials. Yes, to get energy. Very good. Now tell me where energy is present in the body. Yes, every living organism is made up of cells. So every cell need energy. For that purpose, we are taking the different types of the food materials. Yes. Now tell me, what is the chemical nature of the energy? Yes, that is ATP. Chemically, energy means adenosine triphosphate. Now, where this energy ATP is synthesized in the cells, that is, I am talking about the powerhouse of the cell. What is that? Yes, that is mitochondria. So mitochondria prepares the ATP in the cells. But we are taking this much of a huge quantity and macromolecules here every day into our digestive tract. These complex molecules must be reached the smallest cells of the body. Now think how these largest molecules are entering into the cell. Yes, it is very clear that these complex molecules, whatever you are seeing on the slide as a food materials, every food material, whatever we are taking into our digestive system, must containing all these macronutrients, that is carbohydrates, proteins, and lipids, also called fats. These macronutrients, they are converted into smallest molecules, and these smallest molecules through blood, they are transporting to the cell. And in the cell, we are getting energy by the action of mitochondria. Okay, so it is very clear now. Digestion is most important. Now, in the physiology of digestion, in this session, you have to know about what is digestion, where digestion takes place, what is the need of digestion, where digestion starts and completes, and lastly, what are the end products of the digestion. Okay, students? Okay, now coming to the digestion definition, the conversion of a complex non-diffusible food substances into simple diffusible forms. This is the definition of digestion. This digestion can be in the form of two ways. One is mechanical process and a chemical process. In a mechanical process, the food is digested or breaks into smallest pieces here. Cutting, chewing of the food material comes under the mechanical process. Whereas the enzymes, different types of enzymes are present in the digestive tract, and those enzymes convert the largest carbohydrates, proteins, and fats into their diffusible forms. That is called chemical process. Now, look at this video here. We have to concentrate how the food materials are transporting from the one part to the another part in the digestive tract, starting with the mouth, buccal cavity, pharynx, esophagus, stomach, and from the stomach, the food enters into the first part of the small intestine, duodenum, then the small intestine. After digestion, undigested food is converted into the fecal matter, and that is moved into the large intestine, and lastly discharges to the outside. Clear? Now, in the physiology of digestion, we are converted this topic into three major headings. That is digestion in the buccal cavity, that is what is happening in the buccal cavity, and digestion in the stomach, and lastly digestion in the small intestine. Now we discuss one by one. First with digestion in the buccal cavity. Already you know very well about the buccal cavity. Yes. So the space between upper jaw and lower jaw, this is called buccal cavity. Buccal cavity performs two major functions. Look here, one is mastication, second one deglutition. Nothing but it is also called swallowing. Mastication. What is mastication? Look at this video. Here, the food material entering into the mouth, buccal cavity, and teeth are making smallest pieces, and you also observe about the oozing of the saliva into the buccal cavity from the posterior part of the buccal cavity, and also sublingual part, that is below the tongue, the saliva is oozing into the buccal cavity. That is the mixing of the food material, cutting of the food material, and chewing, all are collectively called mastication. So in the buccal cavity, look here below the tongue, the saliva is oozing into the buccal cavity and mixing with the food material. After mixing with the food material, that food bolus is entering into the pharynx and then esophagus. Look here in this, you have to concentrate how the saliva is oozing into the buccal cavity. Look here, saliva is entering into the buccal cavity from the salivary glands and mixing with the food. So the action of saliva in this session, saliva containing mucus, electrolytes, and enzymes. The food present in the buccal cavity is mixed with these three substances present in the saliva. One is mucus, second is electrolytes, and third is enzymes. Mucus for the lubrication of the bolus, that is easy movement of the food bolus entering into the pharynx and then slowly slipping into the esophagus, that is called lubrication. And electrolytes also present in the saliva. Those are sodium ions, potassium ions, chlorine, and bicarbonate ions. Now coming to the important part in the saliva, that is enzymatic action. The enzymes present in the saliva are salivary amylase and lysozyme enzyme. Salivary amylase, also called as tyalin, you know very well, and another enzyme is lysozyme enzyme. Look here, the digestion starts from the buccal cavity because 30% of the carbohydrates are digested in the oral cavity, also called buccal cavity, in the action of salivary amylase. Salivary amylase converts the carbohydrate starch into maltose sugars. That means digestion starting from the buccal cavity, and you have to chew the food material. So whatever the food you are taking into the mouth, you have to chew properly and mix with the saliva, and then maximum maltose is converted. That means when you have taking the food material, don't swallow as it is without chewing or cutting. So many students and the people I heard and seen, they are taking the food material into the mouth and directly they swallow, and they tell nothing will happen. There is no time to me to masticate. No, it's the bad habit. From now onwards, you have to chew in the mouth the maximum time, because it makes the maltose and also the maximum absorption from the small intestine. You have to masticate food material maximum in the buccal cavity itself. Okay, students. So here, one point here, digestion starts in the buccal cavity by the action of salivary amylase. How much amount here? 30% of the carbohydrates are digesting in the buccal cavity. And the pH of the saliva is 6.8, slightly acidic in nature. Salivary amylase works in the pH of 6.8, and the starch is converting into maltose. And another enzyme here, lysozyme. Lysozyme is the antibacterial enzyme. This enzyme kills the bacteria present in the food molecules. That is called antibacterial agent. Okay, students. So up to here, this is called the digestion in the buccal cavity. Only carbohydrates are converting into the maltose. Next session here, digestion in the stomach. Look here in this video, you are seeing the bag-like structure. This is stomach. These are the impulses from the brain and the stomach to the brain. And this is the food bolus from the buccal cavity, and then pharynx and the esophagus, the food material bolus entering into the stomach. Look here, the liquid present in the stomach, this is called gastric juice. The bolus from the esophagus is mixing with the gastric juice. And already told to you, mechanical digestion also takes place. That is, the stomach shows involuntary movements. Look here in this video, you are seeing here the movement of the stomach. See, this is the one of the involuntary movement. Means stomach always makes involuntary movements for the proper mixing of the food material in the gastric juice. And after the mixing of the this food material in the stomach, the food enters into the next part, that is called small intestine, by a valve here. This is called pyloric sphincter. So pyloric sphincter allows only small amount of the food into the small intestine. So this is the about the stomach. Look here, stomach containing the three major important HCl, prorenin, and pepsinogen enzymes. So here one acid and two enzymes are present in the gastric juice. And whatever the food present in the stomach, that is called chyme. Chyme means the semi-solid jelly-like material present in the stomach gastric juice, that is called chyme. And now we are entering into the how this HCl, prorenin, and pepsinogen act on the food material in the stomach. Now, the gastric juice containing HCl. This HCl provides acidic medium in the food material, that is 1.8. The pH of the gastric juice is very much acidic, that is 1.8, because the pepsin, an enzyme present in the stomach, it acts only in the 1.8 pH. And now here you are seeing about the prorenin and pepsinogen. This pro and gen, whenever you are encountering this type of pro and gen words, you have to know about these are the inactive enzymes. Means inactive enzymes first converted into their active form, then these active enzymes act on the food material. For that activation purpose, HCl present in the gastric juice that converts the inactive enzymes into the active enzymes. So now the active enzymes in the stomach are renin and pepsin. These two are the enzymes. You know that renin is one of the enzyme in the gastric juice of the infants that converts the milk into curd. And pepsin is the enzyme converts proteins into proteases and peptones. So this is about the action of enzymes in the gastric juice. In the examination also, we have to write like this. We have a question like, describe the digestion of proteins in the stomach. 4-mark question. First, you have to write about the stomach and what is present in the stomach: HCl, prorenin, pepsinogen, the acidic value of the stomach 1.8. Then the inactive enzymes are converting into active. Then you have to show about this enzymatic action in the reaction mode. Look here, prorenin is inactive form in the presence of HCl converting into the renin active form. Okay. Next, pepsinogen is also inactive form converted into its active pepsin in the presence of HCl. Once this pepsin is created in the stomach, immediately this pepsin converts remaining pepsinogen into pepsin also. This is called autocatalytic activity of the pepsin. Okay. Now, the active form of the renin act on the milk protein. This is the casein. Casein is the milk protein. This milk protein, in the presence of the renin enzyme and calcium ions, it is converted into calcium paracaseinate. This is called curd. This calcium paracaseinate, in the presence of the pepsin enzyme, converted into their peptones. Next, whatever the proteins are present in the gastric chyme, those proteins are converted into proteases and peptones in the presence of pepsin. So this is the enzymatic action of gastric juice enzymes in the stomach. So in the examination also, we have to show in the reaction model also. Okay, now up to now we completed about the digestion in the stomach is completed. Already I stated the physiology of digestion. In this session, we are divided into 3 parts. The digestion in the buccal cavity, second, the digestion in the stomach, and third one is the digestion in the small intestine. Now, in the small intestine, look here, small intestine is divided into 3 regions. The first part of the small intestine is called duodenum. The middle part is called jejunum, and the last part, posterior part of the small intestine is called ileum. Coming to the first part of the small intestine, here, look here in the diagram, here, this you have to see this U-shaped tube here. This tube is called duodenum. Duodenum receives 2 important juices. One from the liver, that is called bile juice. Another important juice from the pancreas, that is called pancreatic juice. So bile duct from the gallbladder and pancreatic duct from the pancreas, both are opens into the duodenum. In the duodenum, these two juices mixed with the food material which is coming from the stomach. Okay, children, now look here. Digestion in the small intestine, receiving the bile juice from the liver and pancreatic juice from the pancreas. Another important thing here, that is the pH of the stomach juice, that is 7.8. Here the intestine also secretes some juices. So here liver, bile juice, pancreas, pancreatic juice, and third one intestine, that is called small intestine, secretes intestinal juice. That intestinal juice collectively called succus entericus. The pH of succus entericus is 7.8. Okay, children. Now, how these succus entericus and the bile juice and the pancreatic juice, how they act on the food material, now we will discuss. And another important thing here, digestion completes in the small intestine. Okay, from the first bile juice, next pancreatic juice, third one succus entericus. In this style we have to discuss now. Now, digestion of the fats. Look here, the fat molecules are converted into emulsified fats itself by the action of bile juice. Bile juice from the liver act on the fats and convert into the emulsified fats. Except this function, bile juice cannot function or act on the food material for digestion because there is no enzymes in the bile juice. So these bile salts, they convert the fats into emulsified fats. What is meaning of emulsification? Largest fat molecules are converted into the smallest droplets of the fats. That is called micelles of the fats. That is called emulsification. After emulsification, these fats are converted into the fatty acid and glycerols. So here the fats are emulsified in the presence of bile juice. And bile juice, here look here, second point, bile juice also activates lipases of pancreatic juice. The pancreatic lipase also called as steapsin. Okay, now the pancreatic juice. The pancreatic juice from the pancreas contains the following enzymes. Look here, amylase. In the buccal cavity, we discussed about the salivary amylase. Both are same. Both are act on the starch and converts their starch into the maltose. Here, amylase is the pancreatic amylase is converting the starch into their disaccharides, and trypsin, chymotrypsin, carboxypeptidases, aminopeptidases, nucleases, all these are acting on the proteins. Nucleases are acting on the nucleic acids, that is the DNA and RNA. And lastly, lipase, an enzyme, acts on the lipids. And how these enzymes act, we will discuss one by one. Look here, proteins, proteases, peptones. These are converting into tripeptides and dipeptides. Proteins, proteases, peptones are converted into tripeptides and dipeptides in the presence of trypsin, chymotrypsin, carboxypeptidase, aminopeptidase. So here, what are tripeptides? What is a peptide? Peptide is a bond between two amino acids. Already you know very well that proteins are made up of amino acids. A long chain of amino acid is called a protein. So here, peptide is the bond between two amino acids. Tripeptide means three peptide bonds among the four amino acids. Dipeptide, two peptide bonds among three amino acids. So small chains of polypeptides, the small chains are creating here. They are with four amino acids and three amino acids. Next, the lipase present in the pancreatic juice converting these emulsified fats into diglycerides and then to the monoglycerides. These monoglycerides once again are further converted into their end products as fatty acids and glycerol. So fatty acid and glycerol are the end products of the fats. So emulsified fats from the bile juice, then the action of lipase present in the pancreatic juice is converting emulsified fats into fatty acids and glycerol. Proteins are converting into tripeptides and dipeptides by pancreatic juice. Next, intestinal juice. In the intestine, the juice is also known as succus entericus. So succus entericus contains inactive enzymes and active enzymes also. Look here, peptidases, maltases, sucrase, lactases, lipases, and nucleotidases all are present in the succus entericus. We are discussing about the digestion in the small intestine. Small intestine receiving bile juice, pancreatic juice, and intestinal juice. These three juices are acting on the food material and completes the digestion in the small intestine. We are now discussing about the succus entericus. One by one, look here, trypsinogen converted into its active form trypsin. Enzyme here, enterokinase. Enterokinase is the active enzyme present in the succus entericus. Once the trypsin is created, this trypsin converts remaining trypsinogen into trypsin. This is also known as autocatalytic activity of the trypsin. Trypsin also converts chymotrypsinogen into chymotrypsin also. So this is about the trypsinogen inactive form converted into the trypsin. Now, action of succus entericus on peptides. Whatever the peptides are created, that is the tripeptides and dipeptides, by the action of tripeptidases, the tripeptides converted into dipeptides and amino acids. Whatever the dipeptides are there, they are converted into amino acids in the presence of dipeptidases enzyme. So amino acids are the end products of the protein digestion. Now, digestion of carbohydrates. The enzyme I told here already, amylase, is present in the pancreatic juice. This amylase converts polysaccharide, that is carbohydrates, starch into maltose, sucrose, lactose. These maltose, sucrose, lactoses are called disaccharides. Maltose, in the presence of maltase enzyme, converted into two glucose molecules because maltose is made up of two glucose monomers. Next, sucrose. In the presence of sucrase, also called invertase, converted into one glucose and fructose because sucrose is made up of one glucose monomer and fructose monomer. Lastly, lactose. The milk sugar is called lactose. This lactose, in the presence of lactase an enzyme, converted into glucose and galactose. This is about the digestion of carbohydrates in the small intestine. So here glucose, fructose, galactose, these are called end products of carbohydrates in the small intestine. Up to now, we discussed about the digestion in the buccal cavity, the digestion in the stomach, and The digestion in the small intestine, we discussed about up to the digestion of carbohydrates. Next, the food material also containing nucleic acids. How they are containing this nucleic acid, children? The food material we are taking, it may be plant source or animal source. So plant or animals are made up of cells. By the action of the enzymes, cells breaks and the nucleic acids are released into the food material. That's why nucleic acids also digested in our small intestine. What are the nucleic acids? DNA and RNA are called nucleic acids. And for the digestion of the DNA, there are DNAs. For the digestion of RNA, there are RNAs or enzymes are present. Look here, a small flow chart is here. Nucleic acids are DNA and RNA. DNA and RNA are made up of nucleotides. That is, nucleotides are the building blocks of the DNA, DNA and RNA. Nucleotides means every nucleotide is made up of three components. Look here, one phosphate, one nitrogen base, one sugar, collectively called a nucleotide. Nitrogen base and sugar, only these two are called nucleosides. That means the nucleic acids in the stomach or small intestine, they are converted into nucleotides, then the nucleosides, and then converted into nitrogen base and sugar. Look here in here the action of nucleases. Nucleases are the enzymes act on the nucleus or DNA and RNA. Look here, nucleic acids are DNA and RNA. They are converted into nucleotides. These nucleotides, in the presence of nucleotidases, and converted into nucleosides and phosphate. Nucleosides are further converted into sugars and bases in the presence of nucleosidases enzyme. So sugars and bases are the end products of nucleic acid digestion. So far, we discussed about the digestion of carbohydrates, proteins, and fats. And what are the end products of these food materials? Look here, the end products of digestion. Carbohydrates converted into glucose, fructose, and galactose. Proteins converted into their diffusible smallest molecules amino acids. Lipids are converted into fatty acids and glycerol, and nucleic acids are converted into sugars and bases. Here, lipids are converted into fatty acid and glycerol. Nucleic acids are converted into sugars and bases. So, glucose, fructose, galactose, amino acids, fatty acids, glycerol, sugars, and bases, all these molecules are called end products of digestion, also called digested food. This digested food is very much small in nature, hence they enters into the lacteals or the villi of the small intestine. From that villi, they enters into the blood. Through blood, these digested food enters or transported to every cell in the cell mitochondria. They release the energy. Okay, students. So far we discussed about the physiology of digestion. I think you understood. And the questions, important questions for IPE, that is, first question is, describe the digestion of proteins in the stomach. Or second question like this, describe the digestion of food materials in the small intestine. You have to write about the whatever we discussed now, and also you have to mention the reaction type of writing. Okay, children, thank you for this video.
Sir, thank you, sir. Yatish, sir. Students,
Thank you,
today
madam.
we have completed like all these sessions like about the anatomy in digestion. We have completed the main structure as well as the associated glands and also the digestion part. Now, in the next sessions, we'll be covering how the assimilation will be done. Okay, so stay home, stay tuned to your, sorry, stay tuned to your YouTube channel. Okay, good afternoon, children. Thank you, sir.
ఎట్లా ఆయన ఎందుకు వెళ్ళిపోయారు?
Dear participants, we have come to an end of this session, Physiology of Digestion System. Thank you all for joining us. Stay tuned for more upcoming sessions. Thank you.